Hello everyone, this is our group member.
By 2050, the world population will come close to 10 billion people, and at the same time, the issue of hunger and lack of nutrition already exists at an alarming rate. Other challenges include changes in global climate, limited farmlands, pests and diseases, as well as the COVID-19 pandemic has deeply affected the global food security. In Malaysia, the current agricultural model cannot meet the demands of ensuring food security and sustainable agriculture development of most plant-based food crops. For example, rice production has only achieved 70% of the self-sufficiency rate and is still dependent on imported rice from Vietnam and Thailand. Medicinal plants are also facing the same problem. Currently, medicinal plants and herbs are a globally valuable source for development of new pharmaceutical drugs. However, due to the increase in human population and plant consumption, about 15,000 species of medicinal plants are facing extinction due to overharvesting and habitat destruction. Therefore, developing a sustainable agriculture with the advancement of new technologies should be taken to overcome this global crisis.
Next is how micropropagation can help to overcome the crops and medicinal plant industry. As we know, medicinal crop is one of the most lowest commonly known area in crop production. So, micropropagation helps to improve the yield in functional food plant. There are three ways on how it works. Firstly, the production of small therapeutic extract (STM), which the natural compound of the plant was extracted as template for the precursor for the semi-synthesis. For example, paclitaxel, artemisinin, cannabinoids, galantamine. Second, standardized therapeutic molecule (STE). It was being micropropagated to ensure stability of biomass supply of phytomedicine crops and also maintain genetic consistency. For example, the botanical drug, herbal remedies and also medicines. And lastly, the large therapeutic molecules, which is LTM, helps in the production of large molecules such as protein and polysaccharides with the goal of producing drugs or vaccine at low cost. For example, bioactive Momordica, anti-HIV for Momordica carantia.
First example of micropropagation is for endangered medical tree Prunus africana. Prunus africana had a major problem where their seed takes long time to micropropagate caused by flowering cycle and recalcitrant seed. Based on Comarcage in 2020, they reported that micropropagate induced from nodal segment could fasten the growth of Prunus africana. The stem nodal segment was subcultured in WPM media and WPM with vitamin and sucrose to observe cytokinic effect on axillary shoot, initiation and growth rates. After 6 weeks of culture, the plantlets also undergo acclimatization to harden their ex vitro survivability. The plantlet were put in a small pot containing 2:1 ratio of perlite soil mix and left grow for another 2 months in greenhouse. The second example is photoautotrophic micropropagation of rhododendron. It is a photoautotrophic micropropagation of woody plant like coffee, eucalyptus and acacia for conventional purpose using no-sugar medium in controlled environment. In the study, rhododendron is an ornamental wood species was observed as it shows low multiplication ratio and percentage of rooting in vitro and ex vitro survival. Conventionally, rhododendron were subcultured in a IAA medium to induce shoot during multiplication stage. Then the quarter strength medium with IBA was used to induce root formation during preparation stage. This was to prepare the plant for ex vitro survivability.
There are several advantages to using the tissue culture process. We already mentioned its effectiveness in helping developing countries to increase food production, but what are some other advantages that may be relevant to us? First, the new plantlets can be grown in a short amount of time, and only a small amount of initial plant tissue is required. Then, the new plantlets and plants are more likely to be free of viruses and disease. However, micropropagation may not be the most efficient method for growing plants. The conditions that limit its application include the cost of labor could comprise between 50 and 69% of operating expenses. Then, it demands greater skill in handling and maintenance than conventional techniques. Other than that, shoot-tip derived plant may show genetic instability.